this hypothetical experiment, I'm supposed to be working with my favorite eukaryotic cells. Well, my favorite eukaryotic cells happen to belong to creatures such as cats, dairy cattle, mice, and humans. Only one of these is a typical test subject in laboratory studies, but I happen to own a few mice and I like them very much. I would not run tests on my mouses. But one thing I do not have a problem experimenting with is bread. So, the cells that we will be hypothetically disabling gene A in will be yeast. We will be using either RNA interference or clustered regulatory interspaced palindromic repeats, otherwise known as CRISPR, to limit or completely stop the production of a protein coded for by hypothetical gene A. Since we don't know what gene A controls in yeast, we are hoping to see its lack of effect when we bake bread with genetically modified yeast, such as this flop bread or an interesting pattern. But which of these methods is the optimal approach to use in our yeast? RNAi is a naturally occurring regulatory process that occurs in eukaryotic cells. The process begins with either very small portions of RNA called microRNA, or with double-stranded RNA known as small interfering RNAs. Both are cleaved into smaller pieces by an enzyme known as dicer and combined with a protein to form an RNA-induced silencing complex, or RISC, which attaches to a certain specific section of the mRNA. The differences are that microRNA is transcribed from a gene to take effect on a different gene and works to inhibit translation of the mRNA. siRNA works on its own genes, and the goal in this case is to cleave and degrade the mRNA. CRISPR is a technique of manipulating genes that was developed based on the prokaryotic defense mechanism against invading bacteriophage DNA. The phage's DNA, in a sense, is copied and used against it by the bacterial's DNA and a CAS protein. A section of DNA from the phage's DNA, a section of RNA from the phage's DNA is attached to the CAS protein, and this complex binds to the foreign DNA. Because the DNA is a perfect match to the RNA, the protein can bind them and then cleave the invading DNA, rendering it useless. Is it valid to choose CRISPR just because it's the cooler process of the two? Probably not. So I will instead say that it is my chosen method because it can be used to target a very specific spot on the genome.